Good afternoon. Welcome to this afternoon's Repartee. My name is John Robinson. I'm one of the participants in this discussion. If you don't know me, just a quick introduction. I'm a partner at a firm called Mazarine Ventures. We are technology investors in innovations that address some aspect of water or wastewater, either societal or industry. This Repartee's focus is on monitoring water quality using technology and innovations in DNA and RNA. This is an area that we, Mazarine Ventures, has had our eye on for quite some time. And in the past year or so, we've come across some individuals and some companies that have been innovating around DNA and RNA as it relates to water and wastewater. And each of these guests in this Repartee has a different angle on it. Not all of them are pure play DNA and RNA, but I will allow them to do a quick introduction of themselves, and then we'll get into our conversation. A couple housekeeping issues. There is a chat. There's a chat element, a chat function within this Repartee. So if you have any questions for any of the participants, you're welcome to use that. Good. Without further ado, I'd like to go around the horn and offer each individual an opportunity to introduce themselves. John Higley, since you're sort of on my screen, you're right next to me, maybe you could go ahead and just provide a quick introduction to yourself and your company.
Yeah, sure. Hey, everybody, I'm John. My background is as a molecular biologist. My first company IPO'd and I was there doing cancer research, so mostly biopharmaceutical development for targeting cancers and not hitting anything else in the body. We kind of leveraged a lot of those technologies that I used in there and at another company in early stage cancer diagnostics to create ECO, which is a company that leverages biotechnology to kind of monitor and address invasive species problems and endangered species problems in the environment. We work exclusively with DNA and RNA. RNA is kind of what sets us apart from a lot of the rest of the field and allows us to really give actionable information to the consumer. And so as far as who I am, I've been a molecular biologist for about 15 years. I've done all kinds of fun things, and if I get a little sweaty or red, forgive me. I'm in Austin, Texas. It's 102 outside, and I have 4 kids and 3 dogs, and they're all inside. So I have the outdoor office with the misters too. So that's kind of my quick and dirty, and I'll keep the ball moving.
Good. Thanks. Khalid?
Hey, everybody. My name is Khalid. I'm the co-founder and CEO of SimpleLab. We're an early stage company developing biosensors for public health. My background is in biochemistry. I trained as a biochemist and got my PhD from the University of Missouri in 2016. I followed that up with a postdoctoral fellowship at Northwestern University Center for Synthetic Biology, where I led the development of a cell-free biosensor platform. So we were really thinking about how do we turn to the environment, figure out how microbes are solving the sensing problem, and repurpose those into really simple reactions that we can freeze dry, we can embed in paper, to really decentralize and democratize water quality testing. Prior to COVID-19, we were focused largely on heavy metal contaminants, but we have since pivoted to developing a COVID-19 assay specifically for environmental detection with a focus on wastewater as well. So thanks for having us.
Great, thanks. Take?
Hi, John. Thanks for having me. Take Ogawa, CEO and co-founder of Veriset. We're a company that's particularly focused on the microbiome, which most people define as a community of microbes, have it be bacteria, virus, or fungus, what have you, that live in a particular environmental niche. And it's probably the most popular for the human gut microbiome and probiotics and so on. But we take that same science and that same approach to look at fecal source tracking. So as most of us know, in surface waters such as beaches and rivers, we are constantly at risk of contamination or pollution based on those types of pollutants. Obviously, there's many other types of pollutants, but we, again, focus primarily on fecal pollution. And we have a novel technology around that. It's a microarray technology called the phylochip. Basically, the way this works is most other genomics tools do is we take a holistic approach of we don't assume what's in the particular environment in terms of which microbes are there. And that's obviously the advantage of DNA is all living things have DNA or RNA in case of some viruses, as we're all quickly learning here in the last couple months. So we're able to take that information from a particular sample and extrapolate, or rather analyze all the bacteria that live in that particular sample, and thus not only tell you if there are good or bad bacteria there, but perhaps more importantly, where did that bacteria come from? Did it in fact come from a fecal source? Did it come from an animal? What have you? So we have a whole analysis pipeline around that. So our business model is very much a laboratory service model, a fee-for-service model. Myself, I've been in the genomics tools technology space for nearly 2 decades across various companies. Some you're probably familiar with, some that you may have never heard of. But again, I'm immersed on the technology side, and so are my co-founders. And again, we're excited to be here and see how we can help in the public health aspect of water quality testing.
Great, thanks, Taki. Johnny?
Yeah, my name is John Pujol. I'm the CEO of a company called SimpleLab. We are a platform of environmental testing labs and devices around the country. Vision is to make environmental testing, specifically water testing, a lot easier, faster, and more affordable. So on one hand, we've onboarded more than 70 labs across the country to do all kinds of testing, from bacterial, radiological, chemical, drugs and pharma, whatever gets into the environment, we want to be able to test it. And then on the other side of our platform are a large number of users, people at home, commercial businesses, maybe one day water treatment companies and utilities, which need and do a lot of environmental testing. And what we do is we make that environmental testing much easier by giving them a platform whereby they can design their own test panels, order tests, get reports back that make sense, that are easy to understand, and analyze a lot of environmental data all in one place. So it's called SimpleLab, and also, like everyone else, excited to be here.
So, Johnny, you've changed your name recently, right? From Simple Water to SimpleLab?
Yeah, Simple Water to SimpleLab. The main reason is, the truth is, our belief is that lab testing and diagnostics in general are grossly underutilized. It's been stuck in an ivory tower, demand structure, policy-driven, regulatory-driven demand. But the world is changing. I think more people, more businesses, more products want to utilize labs to get answers directly. And so we started with Simple Water because, after Flint and so forth, water testing was a huge issue, still is an enormous issue, but it's just the tip of the iceberg. There are labs of all sorts under the classification of environmental testing lab, food testing labs even, which perform vital services to all sorts of businesses and people. And so we wanted to build a brand around those labs and really elevate the ability of people to utilize labs no matter who they are and what their background is.
Thanks, Johnny. So just getting right into it. So Khalid, you're not utilizing you're bringing the lab to the field, and by printing a biosensor on a piece of paper, essentially. Can you elaborate a little bit about what tool you're putting in people's hands, and how does that help? Well, 2 things for water. First, start with COVID, and then how does that play out into water quality?
Yeah, so certainly our vision is to enable anyone, anywhere to sense the world around them. And we think it makes a lot of sense to do this for contaminants that people are concerned of. So metals, for example, as well as for COVID-19. Specifically for COVID, you have a virus that's encoded in RNA. And the typical, the gold standard medical diagnostics test is known as reverse transcription quantitative PCR. It's a pretty involved process. It involves a thermocycler that cycles through some pretty extreme temperature requirements, and that prevents it from, scaling, right? And I think this is the challenge that we saw throughout March and April, was that we were just not able to scale demand for diagnostic testing. But it also prevents it from being used out in the field. If you have to do this quantitative reaction on a, $25,000 piece of equipment that requires, a master's or, a lot of laboratory experience to operate. So We're working on a very simple isothermal amplification reaction. So this functions at a single temperature, and we're doing it at just above ambient temperature. So the power requirements for the device aren't so extreme. And we're also trying to figure out how to make this, happen in just 1 or 2 steps. So the laboratory process for medical grade testing is pretty complicated. You go through this process of isolating the viral RNA before you can actually do the test, and it's pretty complicated. Thankfully, sort of the environmental space is a little bit more forgiving than the medical space, and so there are other ways that we can quickly isolate the RNA for a test. So that's sort of, I hope that answers your question, but that's what we're working on, is an easier test that can be implemented on site with minimal expertise.
Got it. So John Higley, because we have 3 Johns on the call. So what is your approach, or what is your innovation around buffering the RNA in the field and getting it back to the lab?
Yeah, sure. So we're interested in slightly different stuff than what Khalid's doing, but I would like to know kind of which isothermal chemistry are you guys utilizing?
Yeah, so we've got a number of different ones in progress. So we are evaluating a lot of the CRISPR-based technologies, but we're also looking at, RPA is a common one, although it seems to be locked up from an IP perspective, as well as NASBA and rolling circle amplification and all of those.
Okay, yeah, just wondering, just curious, because we were looking to isothermal stuff. We just don't know how much it really gains for us. Essentially, what we're looking at is invasive species and endangered species, and what we've kind of, our secret, we have these automated sampling platforms that get installed in the field. They take the sample, concentrate the sample, and we need to be able to get a broad spectrum. So not just test the water, like a small amount of water. We need to be able to look at like 2,000 liters at a time. So we concentrate that over a series of filters, and then we have a buffer that we've created that's, you could dump it in the lake and it doesn't violate any regulatory standards. So that's that was the difficult part of making something like this. And it's stable at -30 up to, I'd say 80 Celsius. So pretty stable throughout the thing. You don't have viscosity changes. There's a lot of issues when dealing with something out in the environment. And what it does is it stops the degradation of RNA and DNA. It stops the degradation of nucleic acids, so that when we receive the samples in the lab, we can do a lot of complex testing and probing for genes of interest. For we can do the DNA stuff. That's kind of our basic risk analysis platform, right? Like, you can look at DNA trends over time. That tells you if something's growing or not. But DNA is pretty stable in the environment. It sticks around for a long period of time. So it doesn't really tell you if an invasive species like a carp or a zebra mussel is actually there. It just tells you if it was introduced at some point. We've got a lot of data in that range, so that's not really actionable. So we focus on the RNA because that allows us to tell, since RNA breaks down so much more rapidly, it allows us to take a snapshot of alive or is it dead? How robust is the population? Is the population responding to stressors like heat or pH, depending on different genes that we probe for? And is it about to spawn? And so that allows us to offer a lot of really useful and actionable information to our clientele, which are typically managers of either, it ranges on the private side, everybody from like a golf course that takes a lot of water, raw water out of a system, to like a nuclear power plant, municipalities, obviously, and civil engineering firms, and environmental engineering firms. Those are our channel partners. So we kind of work with them to be able to provide monitoring solutions that give actionable information to our clientele, and that's kind of our market and our niche. We're also working now on some endangered species applications, because right now for endangered mussels, the standard practice is to send out a guy with waders and a stick to poke around in mud, or some divers to sift around in the muck, and we can do that a heck of a lot easier by just setting an automated sampler with a pump, programming it to take samples whenever we need it, and then all they do is take the sample bottle, ship it to the lab. It's there's no degradation of RNA or DNA at ambient at temperatures that we've tested from 20 degrees Fahrenheit up to 106 degrees Fahrenheit, because we launch this in Texas, and that was the ranges that we got up in the hill country. So it's pretty stable, works really well, allows us to give real actionable information. So a little bit different than Khalid, but we did look at the different isothermal technologies. And I was thinking about it. I talked to a guy at USGS who I should introduce you to. Good guy. We're out of Wisconsin, and he's doing some isothermal stuff right now that he would be really interested in your technology.
Yeah, the isothermal stuff is a little bit challenging to get the same sort of sensitivity as the gold standard test. I think that's one of the challenges that we're facing. Another sort of really open-ended question, I think, for COVID detection in wastewater is that nobody really knows. We're still trying to learn as much as we can. We don't have a standardized workflow. There's some people that are working with raw wastewater.
Right.
There's some people that are working with process samples. So there's a lot that we have to figure out, but we're hoping that, maybe we can develop an isothermal amplification and at least put together a kit that will address some of the demand, hopefully by the fall.
So, by the way, for everybody who's listening,
that's awesome.
for everyone who's eavesdropping in on our conversation here, the agenda for this repartee is conversational. None of these companies are pitching. There's no presentations. We're just having a conversation, and you're eavesdropping in. If you do have specific questions for anybody, you want to put something out there, you're welcome to use the chat board. Johnny Pujol, so your company is not in the DNA RNA business, but since you're in the testing business, talk a little bit about what your exposure is to DNA and RNA testing, and what your view is on it. Are you starting to see more inquiries for it, and what's your view on this topic?
Yeah, and I mean, I think thanks for saving me the embarrassment of having to say I don't know much about DNA and RNA testing. What we do know a lot about is
okay, Johnny, I like you.
Yeah, thanks, Pugh. So people, one of the things we gather best is questions from the general public. We have several hundred people who call in or message in a day. And most of them are concerned about if it's drinking water, coliform. And that's pretty much all that really matters. I mean, that's the majority of what matters, coliform and E. coli for drinking water. But we have a large number of people, especially this time of year, who start getting worried about recreational waters. We have people who are concerned about their neighbors, and especially on the home inspection front, a lot of septic leakage getting into streams and rivers. And these turn into really violent and ugly confrontations between neighbors, between business neighbors, between residential neighbors. And when everyone just has a coliform test, it's really just not enough to tell the full story of a surface water biology. And there's certainly an opportunity on the residential scale to introduce something more significant, like we do already with isotope tracers and radioisotopes. I think the lack of something comparable for biology is definitely a factor when someone comes to us and asks for, hey, our water has a funny smell or a funny taste. We analyze it. We see a high HPC. We see no E. coli, no coliform. There aren't a ton of options in the market for kind of explaining cost effectively what those what species are in the water. And once you can identify those species, where they're coming from, and solve the problem, from the standpoint of forensics, I'd say that's where we encounter this the most. And a lot of the forensics works ends up being either legal concerns, lawsuits, and stuff like that, or people just trying to get to the root of a problem. Especially in rural America, where you don't necessarily have as much oversight on things like septic leakage and big ag. There are people here at Berkeley who I know are looking more and more closely into the biology of drinking water. It's not just coliform or nothing. There's a bunch of other living stuff there in the water. Those living things generally are harmless, which is why we've gotten away with a fairly simplified approach. But we get calls every day from people saying hey, there's something growing, there's some slime, right? What is this gunk? And it's clearly a living thing, but it's not a coliform bacteria. Telling them it's safe because it's not coliform is not enough of an answer. And so, that's where we encounter this problem a lot, and where I'm excited to see some development. Like low-cost testing could give people answers as to why they have these problems. Right now, they just kind of sit in the abyss without any real answers.
So, Johnny, you said low-cost testing. For the sake of this conversation, low-cost DNA testing, and you mentioned the word forensics. So, if you have to know, DNA is the most accurate signature of life, correct? And so that becomes the best way to make a case against, or to identify who and where is the source of the contamination, right?
I think like that's where where we specialize is not in figuring out the nuances of how these testing methods work, but rather working with professionals to figure out which methods are most appropriate in which situations. And fortunately, the labs and like the other gentleman on the phone here, like they know where their test works and where it doesn't. Mm-hmm. And so what we try to do is say, well, someone comes in to us with even less idea of what's going on, and then we try to match them up with the test they need. So if it's a specialized DNA test or a microarray or an isotherm, to be frank, I don't know the nuances here. But making sure that people with a particular problem are then identified as needing this particular test is what we like to specialize in. There's no doubt about it. It's a very interesting topic. Try telling someone that there's something living in their water, but it's not coliform, so you're fine.
Yeah, so we see this a lot, like in toxic algae. This is kind of a great case study for this because, so with toxic algae, there's lots of ways to detect toxic algae. Everything from PCR to look at the DNA and see what's there. But ultimately, just because the organism's there, that doesn't always translate to how much toxin it's producing. Even if you get down to the subspecies, environmental factors affect how much toxin it's producing. So then you want to go to something like an ELISA or something like that to figure out what the toxin is. But these proteins are so similar that the ELISA isn't super great at telling you how much toxin is there. So the gold standard becomes mass spec. But really, all those different technologies, and I know I just threw a bunch of jargon at people who are maybe listening that aren't scientists, but all those different technologies have a place in the mitigation strategy. All of them are useful, but they're useful for different applications. And so kind of to really fully understand is your drinking water safe, or when is it not going to be safe, and how do we plan for that, and how do we mitigate it? It takes a family. I mean, I used to say this in cancer all the time. People would ask me if well, is this person a competitor because they're making something that attacks the same lymphoma? And no, not at all. And the reason is because you don't go to war with just your infantry, right? It's complementary therapeutics, and the same thing, complementary diagnostics. You have to have a complementary approach is the only one that's going to be effective. And so it's kind of that's why I'm really excited about this conversation, because just from the panel that we have here, everybody has some really cool tech doing some cool stuff. And there's a lot of ways that we can complement each other and solve some big problems in water and water quality and sustainability.
Nice. Well framing, John. I want to bring Taki into this for a second, because Khalid was talking about the tool that they want to get in people's hands to enable the average person to be able to do a test at home, right? But Taki, your Philo chip, you have no intention. It'd be really hard to do that. What is your test, and why is it not able to get that into why can't you get that into someone's hands?
Yeah. Thanks for the question. I like the analogy, the sort of the multi-pronged approach, or the analogy Johnny used around, sort of infantry, and if you go to war, you don't just use it. The analogy that's a good one, but the one we like to use, and this is pretty common in the diagnostic world, which is perhaps not so common in the water testing world, is the difference between a screening test and a diagnostic, right? And we're sort of using forensics and diagnostics interchangeably here. But the analogy we like to use, or perhaps the one that's probably we can all, relate to in one way or another, is the at-home pregnancy test versus an ultrasound.
Right.
At-home pregnancy test is supposed to be accessible. You could go to any Walgreens or CVS and get one. It's cheap, and it gives you a very straightforward, yes/no answer. There's no second tier to it. It's supposed to be a catch-all. That's what all screening tests are supposed to be. It's supposed to be as many false positives as possible, right? That's by design. And the current, on the topic of, fecal contamination or pathogen contamination, as Johnny said, E. coli and Enterococcus, these are indicator bacteria that predicts if there is fecal contamination or not. But as most, if as we think about microbiology, the study of all the various microbes here, we know that E. coli can exist in many different forms and many different niches. Most animals have E. coli in their gut, but not all are dangerous to us. Again, analogous to sort of the algal bloom situation. So where we see ourselves sitting in this whole paradigm is that we're that diagnostic back end. We're going to continue doing screening because it's the most accessible, it's the cheapest, and it's a catch-all. So we'll continue to do these fecal indicator tests like E. coli and Enterococcus and other ones that are out there. But again, that's going to be a catch-all test. When you keep getting a recurring positive signal at a beach or your favorite lake or what have you, well, as Johnny hinted at, we need to know what to do next, because that in itself is not actionable.
Right.
We don't really know if there's a contamination there. Besides, where is it coming from? What's the source? And so that's where we come in. So granted, our technology, like an ultrasound, is relative, it requires expertise. I would say it has to be centralized from a workflow logistical standpoint. I would say the cost, as with most genomics tools, have become very accessible because through miniaturization and scalability, the cost has become accessible. But from a logistical workflow standpoint, it still needs to be centralized. But that's fine because we're that follow-up test, right? We need that deeper expertise, that deeper analysis, that deeper interpretation. So that's where we sit in the paradigm of things, is we're that follow-up to your screening test.
Okay, cool. That's a good answer to that question. I want to get back to Khalid for a second. PFAS has come up in the news all the time, and anybody who's logging into this call knows what that is. We don't have to elaborate on it. What is your take on the difficulty of identification of PFAS in the drinking water? And is that something that is on your near to long-term pipeline of offerings?
Yeah, so it's definitely a long-term, it's a stretch goal, if you might, right? We'd love to be able to offer a simple screening test for PFAS. The difficulty with PFAS and that family of compounds, the reason why it's so nasty is because biology doesn't really know what to do with it, right? And so there's not a lot of biochemistry that is found in nature that can break down these compounds. And similarly, there's not a lot of biochemistry that microorganisms have to detect these compounds. There is some work that I've seen from some group that suggests that there is an enzyme that can potentially break these things down, but it's super early. I think it's probably years, if not decades, before that could potentially be used as a sensor.
The feedback in, is it somebody? Okay, it's gone. All right, so we're at the bottom of the hour. So the first 30 minutes, I've allowed an opportunity for each of the participants to talk a little bit about their technology, their company. I want to spend the next 30 minutes talking about sort of broader trends of DNA and RNA. And the first one, I'm just going to put it out there. So I've kind of gone around and asked people different questions, but I'm going to just put a few questions out there. One just came in. I had a few other questions teed up, but Mark Bertler just put a really nice question out there. He'd like to hear more comments on projected targeted price points to allow scale up of the business. I would assume they all vary. So I guess he's asking each of you what your thoughts are on the targeted price. I mean, maybe I'll just point this towards Johnny, because you're so much in the marketplace of buyers and sellers of water quality testing. Any commentary, Johnny, on the price of a lead test or an E. coli test, or what is the trend, and where do you see prices going in the next few years for a company or an individual who wants to test their water? It's a hard one.
Yeah, hard question. I'll just state the obvious. There's people who have a nearly infinite appetite for diagnostics and the wallet to support that, and there are people who can barely scrape together enough for $25 test. I think in the consumer class, here's the thing: if it's hurting you, you'll pay a lot. If there's if you've got a stink or a taste or a health issue, the dollar price that you'll pay for diagnostics is 10 times higher than somebody who's just curiously browsing around things they can purchase online. Okay, so with that said, where we see where we've done the most biological testing is typically industrial companies that have some weird thing that's going wrong in their factories all of a sudden, or in their product, like a jacuzzi company that all of a sudden is reporting that their last 20 products they shipped out are all foaming like crazy. And they think it's microbial, and we're trying to figure it out. In that spectrum, the companies will spend a lot of money, because once this starts becoming a problem, it usually stays a problem until it's solved. Mm-hmm. And from the standpoint of selling a solution here, that's good news, because it means that there's like a good budget for doing this. Now, if you're talking about trying to sell people a random 23andMe of what's in their water or what's in their pond, I don't think you're going to sell something more than $250. Right. But if there is a discernible organoleptic experience associated with whatever is causing this problem, then really the sky is the limit. We've sold very expensive tests to people who you would never suspect would pay thousands of dollars for testing, because they just need to get to the bottom of it, and there really aren't a lot of solutions out there. And that's one thing I wanted to add earlier, was that what's so important is that these technologies get into the labs, because if the labs don't adopt them, the prices stay stupidly high. Nobody uses them, and there's definitely it's a two-sided market. People will demand something as the price goes down, but the price won't go down until people are demanding it, right? It's like so many industries. In our industry, if the lab doesn't have the knowledge on how to run a particular test, then they're not going to be able to sell it cheaply. Mm-hmm. So getting diagnostics into the labs, or getting those labs more capable of selling that test and putting it online for people and companies to access is actually the biggest blockage that we see.
I want to allow some other people to chime in on this. Mark Bertler, thanks for that question. And one quick follow-up question is, the labs, Johnny, doesn't strike me as the labs are very market forward, or have their finger on the pulse of the market very much. And can anybody else, maybe Higley, do you have thoughts on lab innovation and labs coming into the 21st century?
Yeah, sure. I wasn't trying to comment. I was showing my slides, but outdoors, Texas. What are you gonna do? Yeah, so there's economies of scale here, right? I mean, and we do this in when I worked in diagnostics, you do it all the time. Is most of these technologies that we're all talking about in the lab, there's liquid handlers, there are high throughput machinery. There's ways to make the cost of these go down super duper low. Right now, for a one-off, it depends on how much you're trying to test. So we do high throughput, and our costs are going to come down and our margins are going to go up as we get more business. That's just how it works. In general, the lab space issue, there's a number of issues. Labs are expensive. Having a qPCR machine, and an next-gen sequencer, all the centrifuges and -80 freezers and everything else, it's millions of dollars, and we're very fortunate with our lab. We have a great capability there, but lab scalability in a new market is a big problem for driving down costs. And it's something that the more we get attention to this, and more we band together on this, and the more we kind of create communities where we can have these, communal laboratories, incubator groups, things like that, we can drive down costs further. Right now, we're looking at, my company, I feel free to email me personally. I'll break down all our pricing structure and how we price everything. But ultimately, like every pond, every lake, every river is different. And the problems that we see our customers facing are different. And so hey, we're going to do this kind of sampling this often, and we're going to tell you this information. Yeah, I've got a cookie cutter cost for that. But when it comes to very specific things, it gets a little more complicated, and it kind of becomes more of a conversation. We're working with some group out of Pennsylvania right now to look at lamprey eels in the Great Lakes. And that's a little more complicated. We haven't done that before. Okay, where are we going to survey? Where are we going to put the auto sampler? You need this data to build on that data to figure out the best mitigation strategy to process.
What kind of eels? What kind of eels were those, John?
Oh, lampreys. They're disgusting. I don't know if you guys have ever seen lampreys, but they look like a horror show. They look like something out of another movie. They basically have like a little round mouth full of angry teeth, and they latch onto fish as like a parasite when they're young, and they grow. They're quite long, And they just suck. They're like leeches of the lake, but they're way bigger and scarier, and they're just nasty.
Mhm.
So I can imagine John Higley, because of the nature of your business, invasives are all different, lakes are different, ponds are different. There's some degree of customization of what you're doing. Khalid, you're going the other direction. You don't want customization. You're going mass market. That a COVID test is a COVID test is a COVID test. Tell us a little bit about your product and its universal applicability and your pricing strategy for it.
Yeah, so we're going after the pregnancy test approach, essentially, right? So we don't think our test is going to replace gold standard technologies, and that is not the intention. The intention with all of the biosensors that we're developing is to basically get more testing out there, right? So if we think about our automobiles, we've got like 300 plus sensors on an average car. If we think about our iPhones, right, we have 4 sensors just for motion. If you think about water, what do we have at the consumer level? We have nothing. We might have a sensor on our Brita filter that's basically just a timer. If you have a more sophisticated setup, you might have something like flow rates over time, and that's kind of it. And we know, everybody that's in the water community knows that water is a super dynamic medium. It's constantly changing. If you're looking at heavy metals, for example, the neighbor doing construction or a dump truck rolling down the street can cause your lead levels to spike. And so we're trying to go mass market. We're trying to create sensors that are incredibly easy to use, and that can be used frequently, so that you can start to get more dynamic information regularly. And this is possible because biology is super easy to scale, right? This is the reason why all of these problems exist, is because microbes find a niche in an environment and they start to grow, right? Similarly, we're using the parts from these microorganisms. So we can go to a 500-liter fermenter and grow up enough parts to make millions of biosensors. And we can do things like, using an inkjet printer to print it onto a piece of paper and freeze dry it so that it's shelf stable and that it can be distributed without cold chain. And that's sort of the vision for what we want to do, is to make this as easy to use as a pH test strip would be, or a pregnancy test.
And so let me ask a clarification, just on behalf of the audience. So I would have a packet of these in my kitchen, and then every Monday I use it and then throw it out?
Yeah, that's totally right. Yeah.
And of course it's not a complete test, but it's enough to give me an indicator, yes or no, right?
Yeah, exactly. It's a good screen, a qualitative screen, to know whether or not you're experiencing a problem. And if you are, then of course you turn to our friends on the panel here to do the more in-depth analysis to really pinpoint what's happening.
So here's a question for the group then, because unlike a pregnancy test where you wouldn't really necessarily want to share that data with your neighbor, maybe you do, maybe you don't, but in this case, I could see a lot of value in your neighbor, your neighborhood, your city, if you're getting certain kinds of results, right? So there's a value in the aggregation or sort of the centralization of that data. And we think about the same thing, even from if you're talking about source tracking and you're talking about a perimeter of a lake and there's many houses there, this house probably wants to know if this house is having an issue. Privacy issues, aside for now. I'm wondering, how you guys are thinking about that sort of that aggregation of data, if you will, for the lack of a better term.
Yeah, so we think our screening test can enable the high resolution data collection that we need to start, getting that information at, a community level, for example.
Yeah.
And we're certainly thinking about that. In that case, the customer that's actually purchasing this might not be the consumer, but maybe it's the city of Chicago, because they really want to get to the bottom of the lead service line problem. And instead you're giving it out to, free, and there's 10 tests to every single household. So we're definitely thinking about that, because I think that the actionable information comes from having a ton of data. And, at StemLoop, we're not convinced that laboratory-based testing can get you that amount of data quickly.
Yeah. Never mind. We can move on. I had a thought earlier, but sorry.
One thought that is I think relevant here is that I think what's clear is there's a ton of awesome solutions left and right in this industry, not just biological and DNA, but you look at all kinds of diagnostics in water. It's been a long time coming, but because the industry is so laboratory dependent, so policy driven, like the innovation is so compliance driven, it's practically not driven. So you have this huge buildup of like really awesome stuff. Khalid talks about sensors on iPhones. I mean, like, why are there sensors on iPhones? Well, because there's no one requiring you to put or not put them there. The market wanted them there. We don't have that same force driving innovation in water. And as a result, people have lost confidence in water overall, a fear of tap water, for example. And who's winning in all of this? The food and beverage industry. You're selling tons and tons and tons of bottled water. It skyrockets despite like massive Hollywood and online pressure to stop drinking bottled water. Nobody's listening. It's blowing up still. And I think like the proof is in the pudding in multiple ways. And like what we're seeing is not a lack of solutions, but a kind of demand side lethargy. And we need to find a way to crack through that and make this interesting for people and make it as interesting as iPhones and stuff like that. And it's not simple. It is ultimately your water. It's not going to take pictures of you and your girlfriend at the Grand Canyon. But there is an interesting story to tell there. And what's so exciting about DNA and RNA in particular is that I think this is one of the most interesting stories. It's like the story of life being discovered in either your pond or your yard or your tap water. And it's like that kind of discovery is big. That is interesting and compelling. So I think there's a lot of promise in that to ignite the consumer interest in their environmental data.
Yeah, so Johnny, you bring up a really good point, and this is a question that I sort of have for us as a group. If we think about microbial testing of water, right, all of the standards, which I think we all probably agree are archaic and need to be redone, all of those standards are built around this idea of plating a sample and then counting the number of colonies that pop up 24 hours later.
Yeah.
And we know that, we can use all sorts of technologies like PCR amplification to get a much higher resolution and a more quantitative answer to some of these questions. But, as you say, like, we're not seeing these innovations come to market because it's so driven by this regulatory framework of colony forming units per 100 mils. How do we change that? How do we
you cut their costs? They have regulatory requirements. We all have to play in that, right? So if that's the whole, that's our whole marketing thing is we're replacing 2 guys or 3 people on a boat with a net with a simple thing that you install, and it does it on a regular schedule, and you just all you do is ship a bottle off to a lab, and like it ends up cutting their costs significantly. And we still have that on a subscription service. I know earlier, we usually have to go out and do an initial survey to figure out where to put the automated sampling, but then it's lock and key. It's pretty simple and straightforward and totally scalable. And you mentioned the scalability of biology earlier. It is immensely scalable. And you're talking about printing, growing up in a fermenter. We have a product that we're working on that's it's a protein that specifically kills zebra and quagga mussels and leaves everything else alone. It's kind of based on the work that I used to do in cancer with smart drug stuff. And we're producing it in microalgae. You produce microalgae in a giant pond for 19 cents a ton. That's as scalable as scalable freaking gets, And I think, yes, there's an issue with the consumer not being as interested in water quality as they should be, and the regulatory framework being a hurdle to jump. But if you drop everybody's cost and you provide value within the system, regulatory, we can't wait for it. Okay, we got to find value where the value is. Make your value proposition so damn good that they can't say no to you, and then penetrate the market, right?
My point of view on this is we can innovate and bring testing solutions to market as much as we want, but it's the fear of knowing. From the compliance side, now that I've got this, I got to do something about it. And Johnny, on the home test, we've got lead. What do we do about it? We've got to spend $500 on a solution. I don't even know what kind of solution. I'd rather not even know. And then the COVID test, I don't know. I think people would want to know.
I'm hoping things are changing right now. I mean, I'm hoping people are starting to realize that these esoteric problems that seem like they're just in the ether somewhere when things like a pandemic happen, or climate change down the road, or we see it with zebra mussels all the time. Oh yeah, okay, zebra mussels, and then all of a sudden they're spending millions and millions of dollars a year on mitigation, where they weren't doing that before, and they could have prevented it with a simple test.
Mm-hmm.
It is always harder to sell vitamins than it is aspirins, but it's a necessity.
Yeah. So when we were new to the space and quite naive, we thought utilities and utility operators would actually be interested in our technology. Yeah, turns out we were completely wrong, and it gets to this element of knowledge, right? They don't want to know more information. They only want to do what they're required to do and not anything else. Whereas we've seen that in the private industry, there's a lot of risk mitigation, and they're willing to pay for risk mitigation. Yeah. So if you think about Legionella contamination, nobody wants to be the next Sheraton in Atlanta that was, it settled for $20 or $30 million from an outbreak. And that's what's really driving it. And currently, a lot of that stuff operates outside of the regulatory scope. So I think that's where we've got to focus our value propositions to really drive change throughout the industry.
Taki, I've got a question for you, Taki, about Veriset. Because of the companies here, well, you're all early stage, but Taki, you're still exploring the space. Do you envision going forward that one customer base is forensics and people going after bad actors in the environment? Will that be a source of business going forward? It's not fear of being sued by the government, but it's actually more driven by going after bad actors.
Yeah, I think so. Not to say that, we want to profit from conflict, but in many ways, without a conflict or without an issue, it's hard to justify a diagnostic, right? Like to keep using that analogy, you wouldn't start with an ultrasound. It just doesn't make sense because you're not even sure if you're maybe, it's maybe that's not the best analogy, but that's kind of where we're at. Is we need that unanswered that stagnation action, right? The inaction, because when two parties are at conflict and they both have supporting information and their own agenda, we use science to resolve that, right? And so that's really where, as we've been all talking, we could add value as sort of a surveillance model, sure, but when there's actually an issue, and particularly if it's chronic, season after season, year after year. And what I'm talking about is an example of, where there's some, beach where the local businesses are fighting the residences because they think, there's a leaking septic tank issue or something like that. And they want to install a, multi-million-dollar wastewater treatment plant, for example. Those kinds of conflicts, it cries for solutions like what we're talking about. And so ultimately, that's where we want to be, is addressing problems, addressing conflicts. Now, the bigger question there then is who pays for that, right? Which side? Is it the person that's, suing the other party, the potential, polluter, or the polluter wants to, sort of clean the, get a clean slate? And so that's the experiment that we haven't run yet. But having said all of that, we need to run these experiments of what's the right target market, as it were, and I think that's where Johnny's platform concept, where people come to them knowing there's a comprehensive menu, if you will, of tools and options to answer all sorts of different questions, right? Rather than us, going through the list of, hey, here are all the existing lawsuits in California today, for example. We could do that, but I think there's a different, more scalable way to put the solution out there. So that's, as you can tell, that's sort of where we're at in terms of our exercise here, in terms of how we ultimately market this.
I just want to tag on that. I think every single product here on the screen is like $1 billion problem. The problem is the problem doesn't know what the problem is. And the people who have these issues in their ponds or at their beach or in their well or in their stream, they're like,
right.
And they just, that's the end of it. And because there's no compliance driver, knocking on their door, it just stays put. And there needs to be a kind of awakening of these environmental problems, because they are only going to get worse. And I think this DNA and RNA topic is probably the most exciting one that has emerged that, in the last 10 years that I've, been paying attention. Just because it has that consumer edge, like it's interesting. It's like finding life is interesting. It's much more interesting than finding tin. It has some excitement to it, and there's a story that hasn't been told yet. Like we actually haven't investigated the biology of our waters like we might think we have, and so there's a lot of discovery involved. And I think that discovery will shed light on issues of climate change. It'll shed light on issues of chronic health risks. It'll shed light on waste and industrial pollution. It'll ultimately improve public health and stewardship. But we have to start telling that story in an interesting way.
Mm-hmm. Mm-hmm. Just trying to get the kids out of the office.
No, trying to keep the kid inside. He's letting a dog out.
Johnny, one of the things that we Johnny Pujol and I have talked about this at length is that there's a lot of foundations out there, a lot of successful people have done well, and they want to help, and they want to give their financial resources to a foundation should be granting tests on an annual basis. Just if there's a hurricane in Houston, or if there's a pandemic somewhere, or there's a Flint, one way that wealthy families can help is to just buy tests and donate them. Would that be an accelerant to all of your business? Maybe they just don't know they can do that.
Well, right. I think that's the bigger question, is just lack of awareness, and it's not their fault. It's probably more our fault than anything. But I think there's just a lack of awareness, right? I mean, you're looking at the people on the panel here. We all will love to talk about technology for hours, but ultimately that doesn't matter. How you do it. It's the type of insight, the type of actionability that's more important. Because even for what we do we have competing technologies that we could use? Yeah, of course we do. There's many different ways to measure DNA, for example, to kind of put it loosely there. Mm-hmm. But that doesn't even matter, right? Like we've been talking all along. Most consumers, most people out there don't know this is even a possibility. Most people don't even know that there are thousands of different species of microorganisms that live in a glass of water. They don't know what the significance of that is, and that's not their fault. That's that's our fault.
Sounds like we should collectively start a business that's like promoting. Well, I mean, I'm not going to even finish that, because Johnny, your business is raising awareness, but you're not doing it with fear. You're doing it more about health, right?
I mean, we still wait for people to come to us. We're not out there in any position. What we've gotten good at is like waiting for people to go online looking for answers to these weird esoteric questions their doctor's not going to answer, right? So it's like questions the water utility won't answer, or questions there's no answer to because it's a private well. The people go online and they search, but they've just for better, they've just never been given solid answers before. And so what we're trying to do is formalize and place those questions into bins and say, well, what types of questions are people asking? Most of them are fairly obvious, because you've experienced them yourself. Why does my water smell like rotten eggs? Mm-hmm. You answer that one question, that's hundreds of millions of dollars in answering that question properly for people. People search that question left and right, up and down. They want to know why their water stinks. And as a result, I think because people don't have the language of water quality or environmental diagnostics and lab testing, they don't even begin to actually get close to finding a solution. They just ask a very, very simple question on the internet and get lost in Reddit or Quora or something. Mm-hmm. And before you know it, they're alkalizing their water and bottling it and selling it to their neighbor. And they're just and you don't want to start a business that requires public education campaigns, right? Like that's the problem we're facing. It's like none of us are ever going to afford to teach people this stuff. What we have to do is play with what we've got. And what we've got is people experiencing more and more questions for which there aren't good answers coming from the medical field or from the EPA or from the utility. Mm-hmm. And we have to figure out a way to dial our solutions into those questions, because they actually do answer the questions. Yeah. But we have to play with these simple framings and understand them and respond to them effectively.
And Khalid, thanks, Johnny, for putting it, packaged it up so well. Khalid, you're basically the problem you're solving is just like a simple one. Yes or no? Is it present or not? Is that oversimplification of what you're doing? People just have that simple question: do I have this or do I not?
For our metal, heavy metal sensors, that's exactly what we're trying to the question that we're trying to answer is specifically, are you above what's considered, the safety threshold for that particular contaminant?
So that's solving a big problem for there, because sometimes all people need to know, Johnny, is like, do I have it or not? Or is this a green or a red? I don't need to even know the yellow, right? That's too confusing.
Right.
What about John Higley on your side? Just because we only have a couple minutes left, we'll just go around quickly. What are some of your big milestones in 2020, 2021? Can you share with everybody what your next milestone is?
Sure. So we've already hit our sales goals for the year, which is great. And we're expanding further. We're putting some stuff out in California, and now we're hiring a business development director in the Great Lakes region because, just Johnny mentioned earlier, education is a huge hurdle to deal with. And luckily in the Great Lakes, we don't have that hurdle there for invasive species because they know it, they know the problem. There's a lot of money for it there. So we're hiring a biz dev director there, and we have hit our targets, and we're working on the smart drug technology that we're working on to get rid of zebra and quagga mussels. The data looks wonderful. I mean, we've been out of the lab for a little bit because of the quarantine, but we go back in on Monday, which is great. And so then, that pushed our deadline a little bit with NSF. But yeah, that looks fabulous, and it looks like we're going forward with phase 2, and that'll start doing in-water testing early next year.
So, John, you've got the DX and the RX. You've got the diagnostics and the treatment coming.
Yep, Eco DX and Eco RX. So we do both sides of it. And so the automated samplers addresses everything from risk mitigation with DNA to early stage intervention with kind of one of the RNAs that once you start seeing metabolism crop up, we know what to do, and we actually saved a lake here in Texas doing that. That worked really well. And even with full infestation, we can provide mitigation tactics by looking at how the species are responding to treatment and what treatment and how much is needed.
Cool. Thanks. Hey, John, sorry to cut you off. You got to make some.
No, you're good. I could go on forever. I'm allowed.
Next milestone, honey.
Yeah, so we pivoted about 3 months ago to COVID-19. It's been a great pivot for us. I think it was out of necessity as well as opportunity. We have a lot of inbound interest, some potential pilots in the work. There's a ton of interest, not coming from the utility side of things, but from large Fortune 500 companies that are trying to figure out how to get their facilities back up and running and minimizing their risk from becoming the next, meat packing plant, sort of an outbreak central. So super exciting. We're starting to hire. So if you guys know good synthetic or molecular biologists, send them over to STEMLoop.
Cool. Well, thanks. On behalf of humanity, please focus on the COVID stuff, and when the time allows, make sure that water quality gets back up to on your priority list. Take, next milestone?
I'd say there's 2. We have an ongoing collaboration with several regions in the EPA to basically do proof of concept / validation of our approach to fecal source tracking. So continuing to maintain and move that along. The second part is more on the commercial business side, is to continue exploring some new, call it distribution channels or sales channels, because of all the the various issues that we discussed. And we can only do so much as our small team. So to really continue to explore that, I'll say 2020.
Sorry, Take, I got to cut you off. Get it, Johnny.
Yep.
Johnny, final word. Next step. Oh, you're on mute. You're on mute.
I want to list some DNA and RNA testing on SimpleLab. So that's something we can hopefully do in the next month or 2, which would be really fun. Get some orders flowing.
And what's the website that people who in the audience can go find those tests when they're eventually live?
Oh man, I wish you'd asked me in a week. Go to GoSimpleLab.com in a week. Or you can go to mytapscore.com. Go to mytapscore.com.
We're going to talk. You're going to add our stuff on there too. We already talked about this.
Well, okay. So
I've been the largest website for like 6 months.
I know, we're all freaking busy, man.
So it's funny.
Calling you tomorrow all day.
So, we're just wrapping things up a little bit. So you know, the four of you, I've already connected you guys spiderweb style. So you guys always have conversations going, and so everybody who's eavesdropping on this conversation, if you need anybody's email me. I'm happy to share. Go to their LinkedIn page, follow them. They'll have updates coming through. But if you want Mazarine's 2 cents on any of these companies, we've got notes on all these companies and relationships with all of them, and happy to share as much as possible, which is why we do these things. We did have some questions that came through. I will copy paste these and send these to all the participants so you can, and then who they came from so you can follow up with them. Ohali just put his, actually, if everyone wants to quickly just put their website in here, that would be a creative solution. So I don't have to spend all night sending emails out. And then one more, Johnny. All right, thanks everybody for participating, and to my fellow participants, thanks for taking the time, and looking forward to continuing this conversation. Have a good day.
John.
Yeah, thanks, John. Thanks, everybody.
Thanks.
Thanks
guys.
all.
Thanks, guys.